as I was saying, um, so as the Rubisco has the affinity for carbon dioxide, so they can go through the process to make the food. But enzymes, if you remember back, can be inhibited, can't they? And Rubisco, even though it has affinity for its proper substrate, carbon dioxide, will also allow its active site to lock up or engage with oxygen, right? O2. So O2 is like an inhibitor. If the O2 is bound to the active site of the Rubisco, the carbon dioxide cannot. Now, there's a problem when Rubisco binds to O2. There's a problem when Rubisco switches its allegiance from its normal substrate over to the O2. Okay? None of the rest of this happens. No food is created. This is called photorespiration, and it occurs when plants in dry or semi-arid or arid regions close their stomata so that they don't dry out. The oxygen levels rise, and so the carbon dioxide levels are dropping, oxygen levels are rising. And remember, an enzyme will either attach to a substrate, or the probability, I should say. The probability of an enzyme attaching to a substrate or an inhibitor depends on how many there are. Remember that discussion in enzymes? More inhibitors, better chance it's inhibited. More substrate, better chance it can have a reaction. So as the carbon dioxide levels are dropping in the plant because they're not coming in from the stomata, the oxygen levels are rising in the plant because they're not leaving through the stomata. And Rubisco starts switching over and starts attaching itself to oxygen. Photorespiration. It's called the respiration, photo because of the light, respiration because there's oxygen involved. Right? And nothing happens. Plants can die from photorespiration. Now, does it kill them instantly? No. Plants can have a tolerance for photorespiration, but they're, it's kind of like holding your breath again, right? Eventually, those stomata are going to have to open. Okay, well, other types of plants out there. Now, this is what happens to your typical type of plant. And your typical type of plant is known commonly as what we call a C3 plant. Let me show you where the 3 comes from. We call them a C3 plant because during the Calvin cycle, a lot of the compounds that are made have 3 carbons. Each of those little gray balls is a carbon atom. Not all of them, right? REPP does not. But you're bringing in 3 carbon dioxides. That's why it's called a C3 plant. Now, C3 plants are plants that would live in those areas in which it's dry sometimes, and it rains. It's not arid, it's not semi-arid, it's temperate, right? It's got a mixture of weather. There may be some dry areas and dry times, but they aren't excessive, they aren't frequent, and so plants, really not a big deal. Remember, the thing with plants and water is they got to store it. They can't go get it anywhere. Okay, there's another type of plant out there called a C4 plant. And C4 plants, and another type that we'll talk about known as CAM plants. CAM is an organic acid, CAM. CAM stands for crassulacean. Isn't that nice about science words? They're so phonetic. Crassulacean acid metabolism. Crassulacean acid is an organic acid. All right, now C4 plants and CAM plants are plants that find themselves living in those climates in which drying out becomes an issue. In other words, living in those climates where they're sometimes forced to keep their stomata closed for longer periods of time than C3 plants could survive. So let's take a look at how they get around that. Let's take a look at these adaptations of C4 plants. First of all, the name C4 comes from the fact that during this process I'm going to explain to you, there are some 4 carbon compounds made, oxalacetate, malate. It's just a way to differentiate them apart, okay? Remember, if we look inside the leaf of a plant, what we have are these mesophyll cells, right? And if we look inside of a C4 plant, we have these things called bundle sheath cells in addition to mesophyll cells. Bundle sheath cells do exactly what they say. They're a bundle of cells that make a sheath, a coating around the central vein, the vascular tissue that moves the water and the food and all that stuff around a plant. Just like insulation on a wire, it's a sheath, right? Now, the reason why the C4 plant anatomy is done that way, you can see over on the right what's going on. In order to deal with this problem of photorespiration, what we need to do is make sure that the CO2 level entering or near the Calvin cycle is always very high, because that's where Rubisco is right there. Now, we want to keep the oxygen level low, keep the CO2 level high. Here's how a C4 plant does it. It uses another enzyme, PEP carboxylase, which has a much stronger affinity for carbon dioxide than Rubisco does. So PEP carboxylase is not going to be affected by the rising O2 levels. PEP carboxylase takes the CO2, incorporates it into these 2,4 carbon compounds, which then through these plasmodesmata, right, the openings between plant cells, it basically pumps carbon dioxide down into the bundle sheath cell. All right, and when you pump carbon dioxide, when malate gives off carbon dioxide, it produces pyruvate, same stuff you saw in respiration, which then goes back into the mesophyll cell, turned into this stuff, PEP, which is then used with the enzyme to bind CO2. Now, what goes on down here is while the oxygen levels may be rising in the plant, what we're doing is continually pumping CO2 into the bundle sheath cells, I'll get that right eventually, so that there always is relatively more carbon dioxide than there is oxygen. So even though the oxygen levels might be rising, as long as you've got enough carbon dioxide that's been stored up in this process in the mesophyll cells, you can keep pumping it into the bundle sheath cells, and you can slow down this process, right? You can slow down this process of photorespiration. Again, if you keep your stomata closed for too long, then everybody's going to suffer from photorespiration. You got to get rid of that oxygen eventually. But this is kind of a measure to help you get through those tough times, if you will. However, you think, well, why don't all plants do this? There is a cost, right? There's a little bit of an energy cost. In C3 plants doing it, there's no energy cost, right? But in here, you do have to spend an ATP. Now, of course, you get more energy out of the food you create than the ATP you spend, or it wouldn't happen that way, but that is the trade-off. In order to do this, you're going to have to expend a little bit of energy to drive that reaction in the mesophyll cell so you can always keep that CO2 level pumped up. So that's a C4 plant, like sugarcane. We say that C4 plants have solved this problem of photorespiration by separating the steps spatially. Two different cells, carbon dioxide gets fixed into one of those organic acids, right, which gets dumped into the bundle sheath cells. The crassulacean acid, like pineapple over there, they do it, but they do a little bit differently. They're a solution that's temporal, by time. At night, they open their stomata when dehydration isn't such a big problem at night because the sun's not up, and they bring in carbon dioxide, and they fix it. They turn it into crassulacean acid. That's that organic acid. During the day, the stomata close, right? Sun comes up, starts getting dry, the stomata close, and all of that organic acid that was stored up during the night then starts releasing that carbon dioxide into the Calvin cycle. Again, the oxygen levels may be rising, but since you've stored all this stuff up from the night before, you're going to try to make sure that the carbon dioxide levels are always greater than the oxygen levels. And again, remember, this is a temporary fix. Plants can't photorespire forever. They don't open their stomata, right? If they don't open their stomata, then eventually the O2 levels are going to rise beyond the CO2 levels. Remember, the stomata not only releases the O2, but brings in the CO2. So no matter how much you store or how you store it, you're eventually going to have to open your stomata, okay? Or you die. All right, that's it for chapter 10. Chapter 10.